Hello, my name is Dr. Sterios Moschos and I am an Associate Professor of Cellular and Molecular Sciences at Northumbria University. I'm also the Founder and Chief Scientific Officer of Pulmo Biomed Limited in Northumbria University spin-out. My talk today will be around fractionated exhale breath aqueous particle sampling for diagnostic bioanalytics of COVID-19. I will briefly introduce the concept of exhale breath condensate diagnostics and then introduce to you PBM-HALE, which is a device pictured here on the right that captures fine aerosols separated from large droplets at the point of emission with minimal transit path from the oral cavity. I will give you some data that we have on microbial capture and our other levels of bioanalytics, and we'll explain to you our clinical progress update as we have been working on this for a year now. Exhale breath condensate basically seeks to condense the 40 to 95% hydration level of breath and capture not only the volatile compounds found there, the smells of the garlic you've consumed the night before, for example, but also the biological molecules that are dissolved inside the aerosol droplets in your breath. In the past 20 years, there have been multiple studies that connect about 40 odd diseases, such as lung infections, liver diseases, multiple cancers, even neurological conditions like Parkinson's, with changes in biomarkers found in breath. But the problem has been that, as highlighted in 2008 and 2017 by the American Thoracic Society and European Respiratory Society, that the devices in the market were unable to confirm reproducibility, avoid contamination from saliva or indeed the environment. Sometimes they would capture no sample at all whatsoever, and they would have unclear safety to the healthcare professionals or capacity to separate upper versus deep lung samples. Examples include the ART tube, which has been criticized for its poor process control and lack of salivary contamination removal, and the EchoScreen, which is reportedly unable to yield samples in many cases. And the simple question to clinicians is, have you ever looked inside this elephant tube to see whether or not your sample is just lying in there, condensing on the tube? With this in mind, we started early with some Corning tubes, and we've evolved into this device that you can see on the left. That has a mouthpiece engineered to be shut before use and actuated by inserting a lock and load, as we say, this mouthpiece over a dry ice bucket container which contains your sampling tube. As you action this pulls forward a drawer that opens these two downward pipes into your condensing tube, and then you press down this component here, which is effectively a salivary trap that uses inertial impaction against this wall as you exhale through it to capture the large droplets, whilst the fine aerosols proceed here and encounter the walls of a tube immersed in a cooling liquid, and where we get condensation, thermophoreses, etc. For the fine aerosols. We have used extensively computational flow modeling to basically understand the level of optimization for getting maximal capture of the large droplets in these cavities here, but also to demonstrate that we get efficient cooling of the sample by using this approach. In that process, we've been able to evolve the sampling consistency to a very large degree. So this is an example which is reproduced with multiple cases, where you see highly consistent amount of sample in the fine aerosol fraction between a quarter of a minute and half an hour. And what I need to point out is that the angle of this graph here on the left, which represents the volume of fine aerosol captured over time, will change from individual to individual as the hydration levels of the exhale breath vary dramatically, as we said before, 40 to 95%. We have also been unable to date in any of the fine aerosol samples that we have collected to measure any level of salivary contamination as determined by alpha amylase activity. So this is an enzyme found in saliva that is very active, and even for half an hour's worth of sampling, we just can't detect any of that enzyme. In contrast, the salivary trap contains appreciable levels that are lower but comparable to that of drooling into a tube. If we culture the fine aerosol captured sample, we see here that if you're condensing at very low temperatures like dry ice, minus 100 degrees Celsius, you get no viable bacteria. But if you substitute this with wet ice, you get a level of bacterial culture where you can see big spots here, but also some fungal colonies. And this contrasts to the saliva, which has a very high bacterial load. So exhaled breath contains biomass. It's nowhere near as biomass rich as saliva or some other biological samples that can be used for molecular analytics. But if we try to do what's known as microbiomics, a method of looking at all the DNA from one particular gene that is very variable across the whole bacterial kingdom, and then try to hone down on what bacteria are present, then in this example where we have a mild cough individual, we see a statistically significant above background detection of Streptococcus using this entirely agnostic method of sample capture. Now this method is only good for detecting bacteria, and it's not perfect for telling you what species of bacteria precisely. It can tell you it's strep, for example, but not what kind of strep, and it can't tell you anything about antibiotic resistance. But the DNA is there, so we can definitely analyze that DNA, for example, for antibacterial resistance, antibiotic resistance. But we can also do things like metabolomics. And here you see a list of the compounds that we saw in an exhaled breath constant fraction of one individual. And what you see here is a dominance of these two lipids, which are lipids found in the eukaryotic cell membrane exclusively, which suggests that you have at least fragments or maybe even extracellular vesicles from eukaryotic cells in the airways that are found in the fine aerosol fraction, and they're pretty much dominant in the sample. But you also see known components of the lung surfactant indicative of lower airway components coming out in the fine aerosol fraction. Interestingly, we also detected this fellow here, coscohygrine, which turns out to be a contaminant in poorly cut cheap cocaine, and the individual that offered that particular sample admitted to have nasally abused cocaine 3 days before the sampling process. So perhaps fine aerosol characterization can be used for other things than just clinical diagnostics. But how does this feed into virology? Well, very early in the pandemic, we decided to use pseudoviruses. So this is basically a version of HIV, a membrane-coated virus with a capsid, with an RNA genome, to nebulize it with a machine like this and feed it very quickly through our device. And what you can see here is the amount of fluorescence expressed in cells that have been fed the condensate from the bottom of the tube after 72 hours. So our virus was engineered to express this green fluorescent protein. It was put on the cells, and 72 hours later we looked for that fluorescence. And there's always a little bit of background fluorescence from these cells. But what you can see very clearly is that the condensate contains virus that can infect the cells, introduce that gene, and actually express it. So whilst we can perhaps protect healthcare professionals from bacteria in the breath of patients, we can't necessarily protect them from the virions, but we can certainly capture those virions if they're aerosolized. So effectively, just over 1 virion per minute's worth of sampling, we should be able to capture it very well. We've explored this further, and we've actually been able to determine some of the physical parameters that affect what you measure. So here you see different kinds of nanoparticles, so that's polystyrene beads, liposomes, representing 2 different charge levels, very negative versus mildly negative, comparable to virus-like particles of this HIV type virus, but also SARS-CoV-2 virus-like particles. And what you can see is that the really negatively charged particles basically start to become larger, they aggregate. And as they aggregate, of course, you anticipate the concentration of these viral particles to drop. So we're comparing here the native cell culture liquid after you've frozen it, just simply freezing it, or aerosolizing it and then freezing it again. But when it comes to the viruses, the effect is practically negligible, and it's in the order of loss of infectivity we saw in the other assay. And these results were produced independently in a lab in Germany from our lab. So armed with this information, we set out, before the community was trying to convince everybody that coronavirus was airborne, to find clinicians to work with us to look at whether or not the virus is found in exhaled breath. And we wanted to see only if it's found in tidal breath at the time, because we didn't have the core data or any of that information to inform this process. So we set our study size to N of 60 with our interim data reporting point at N of 30. We analysed the fine aerosols, we intended to collect the large droplets for post-analysis as well, and nasopharyngeal swabs, and we just quantified the virus by RT-PCR. And the results that we have been able to reliably analyse and ensure they've been done correctly tell us that we do not get any false positives, even if individuals who have recovered from COVID and are still in very dirty, poorly ventilated COVID wards are being sampled. So the lung is actually acting like a filter for the air, and it's just basically not allowing viral matter to come out after it's been inserted into the lung if it's present in the rooms. I have to stress, we assume these settings are SARS-CoV-2 airborne positive because there are no ventilation systems. In some cases there was no air conditioning operational, no windows open, etc., and there were a dozen patients in a ward. And none of these samples came back positive. But apart from the standard problems of getting clinicians to work with you on a concept that they thought is wrong, we had additional substantial problems in recruitment. So for example, people just don't turn up to hospitals until they feel really ill, and by then they're in the second week of disease, by which point transmission is much lower. I spoke about ventilation and health and safety problems, where we managed to recruit a centre and had to drop them because of the technical issues there. Healthcare professionals just being afraid of a breath device, they just didn't understand what was going on, which we had to overcome. The capacity of the research team, the clinical research teams, to engage with the research project when we were getting lulls and no recruitment versus surges and no capacity to do research. And of course now we're facing vaccination, but ultimately, whilst we have completed the N of 30 interim point, issues around clinician compliance with the process have basically set us back to square zero. We have to restart the study again. What I can say is that the results that we have to date, although there are technical elements that we need to resolve before we are able to announce exactly what the data is, what we have obtained are fully in line with what was reported from the Swedish group earlier this week, and indeed what has been reported from Don Milton's group early in the year. But we just don't have data with regards to higher level volume of vocalization. But what we can say is that exhaled breath condensate, fine aerosol fractions, and indeed now we're getting data on the large droplets as well, contain bacteria, they contain fungi, they contain cell membrane components, they also have DNA, RNA, protein, and what other funky stuff that might be contributed by the patient, as we saw. And indeed, our device is able to capture airborne virus in the fine aerosol fraction, where it will remain infectious. But because of significant issues with clinical compliance, we can't fully and formally report out on the interim study. We are restarting in Brazil presently, and we have results that indicate that the tidal breath fine aerosol fraction that hasn't had a chance to evolve into fine aerosol from large droplets, salivary droplets, is indeed appearing to be devoid of virus. If you want to find out more about the device SOP and simple explanation how to use it, you can follow these links. Otherwise, I would like to close by thanking everybody in my lab for over the years who has contributed to this project, starting with Amy Wilkinson, who came as an undergrad into my lab and said, I want to go and sample people sleeping under bridges in London for tuberculosis, to John Henderson, who generated a lot of the data, as well as Theodora Manso in the spin-out, who's generating data presently, and the clinicians in Germany, Greece, and soon in Brazil that will be working and have been working on this project. Thank you very much for your attention.